Hello everyone, back with another video with another interesting topic. So the topic today which I am going to talk is on sickle cell anemia. So let us understand about sickle cell anemia today. Sickle cell anemia is a genetic disease with severe symptoms including pain and anemia. The normal hemoglobin is usually oval shaped. Due to the sickle cell anemia, the normal hemoglobin shape will be changed to sickle cell hemoglobin so that it results in clogging in the blood vessels. The disease is caused by the mutated version of the gene that helps make hemoglobin. So, the gene which makes the hemoglobin, the normal DNA, that results in a point mutation, resulting in the formation of defective hemoglobin molecule. So, the hemoglobin molecule, that is a protein that carries oxygen into the RBCs. People with 2 copies of the sickle cell gene have the disease. So let us understand the genetic level. Here you can see the normal gene. This is the normal DNA. Normal DNA is having GAG. This is the normal gene. Normal gene, that is adenine in the middle, adenine, that has been resulted in mutation forming T, that is thymine. So this is a single, that is mutation has occurred here. So due to this point mutation, a single point, the mutation at a single point. So adenine instead of adenine, here there is thymine resulting in sickle cell anemia. In normal, this is the normal DNA. Here you can see the normal DNA having GAG. This is DNA and this is RNA. So in my previous videos, I have dealt with replication, transcription, translation. Likewise, from DNA, it results in transcription. The result of transcription is the formation of RNA. So here you can see RNA GAG. This is the normal resulting in normal protein. So from DNA to RNA, RNA to protein. I have dealt in my previous videos about protein synthesis. So this is how it occurs in the cell. DNA to RNA, RNA to protein. So the normal protein should be glutamic acid. This is the normal protein. Normal gene forming a normal protein, that is glutamic acid. So this is the normal protein, glutamic acid, resulting in normal hemoglobin. But whereas in sickle cell anemia, what happens due to the point mutation, GTG. Here you can see GTG. This is the defective gene, resulting in the formation of defective protein. That is, instead of glutamic acid here, valine is formed. That is a defective protein, that is mutated protein. So this is the defective gene forming this is a DNA, RNA and protein. The protein formed here is defective protein. So the defective protein is valine. The normal protein should have been glutamic acid. Instead of glutamic acid, valine is formed in sickle cell anemia. So what happens is that the normal hemoglobin is oval shaped. Due to this mutation, what happens? Instead of this normal oval shaped hemoglobin, there will be a sickle shape, that is clumped hemoglobin is formed. I will show you the picture. Here you can see this is the normal RBC, that is normal hemoglobin, and here you can see elongated version of here clumped. This is the normal hemoglobin, that is the sickle cell hemoglobin. So here you can see distinguish between the normal hemoglobin and the sickle cell anemia defective protein having sickle cell gene. So what happens is that due to this sickle cell anemia, that is a result of point mutation. A single change in just one nucleotide in the gene for hemoglobin. The mutation causes the hemoglobin in the RBCs to distort to the sickle shape when deoxygenated. So what happens? The normal hemoglobin, the normal protein is glutamic acid. Whereas in sickle cell anemia, instead of glutamic acid, due to the point mutation, valine is formed. That is the defective protein which forms sickle cell anemia. The sickle shaped blood cells, what happens? Those sickle shaped blood cells, they clog into the capillaries, cutting off the circulation. So this is the cause of sickle cell anemia. Valine rather than glutamic acid is formed, resulting in sickle cell anemia. In sickle cell anemia, the gene for beta globin is mutated. As I told in the earlier, that is the gene for the beta globin is mutated due to the single base mutation. Only one base, there is a change resulting in point mutation. What happens? The sixth amino acid in the chain is valine rather than glutamic acid. This is the defect due to point mutation. Some RBCs become sickle shaped and these elongated cells get stuck in the small blood vessels so that the parts of the body don't get oxygen they needed. So this is how the result of sickle cell anemia. Hope this video is useful for most of you. I will be back with more educational videos like this. Till then, take care and bye-bye.